Oh, thank you, Morgan, and hello, everybody. So as Morgan just pointed out, today we will be having two portions of our presentation. I will be starting with the general introduction of CPDC, and Dr. Goodbody will be talking about our OICR projects. So CPDC, or the Center for Probe Development and Commercialization, has been founded in 2008 from the CSER program and has collected so far up to 50 million in grant funding, mostly from the NCE, the Network of Center of Excellence, but also from OICR and a few other granting agencies. As of today, we have about 120 employees in two different locations in Ontario. And I'll be talking about the rest of the points as we go through the presentation. So our headquarters are located on the campus of McMaster University. We have 6 clean rooms dedicated for GMP manufacturing of radiopharmaceuticals. 2 of these clean rooms are licensed by Health Canada, and 2 have been audited by the FDA. So over the years, CPDC has become a globally respected and renowned organization for the GMP manufacturing and distribution of radiopharmaceuticals. So what are radiopharmaceuticals? Basically, they are regular pharmaceuticals that carry a radioactive payload. So these are used for targeted radiotherapies or imaging. So there's an example here with an F18 label CD8 targeting Centyrin, which is a small protein. And basically, radiopharmaceuticals are composed of 3 components. The first is the targeting moiety, which can be, as in this case, a small protein, but it could also be a small molecule, an amino acid, a peptide, or a large protein, such as an antibody. Then you have the linker, and then attached through chelation to the linker, the radioisotope or the radioactive payload. So the choice of radioisotope will depend on the pharmacodynamics property of your targeting moiety and the fact that you are going to have a therapeutic or an imaging agent. So we do manufacture product with radioisotope that has half-life ranging from 1 hour to 8 days. So that brings a lot of challenges because the shelf life of our product varies from a few hours to a maximum of 12 days. So just to illustrate that with the product that we have manufactured for a project in collaboration with UHN, which is the lutetium dotatate. So you can imagine the patient has to travel in many cases to the hospital center to receive his treatment. And then at 7 a.m. on the morning of the treatment, the patients arrive. For lutetium dotatate, there's some pre-treatment procedures, and around 10 a.m., the injection of the drug product is made. In parallel to that, while the patient travels to the hospital, the lutetium isotope travels to CPDC and then start a production that goes overnight with the shipping of the final drug product under quarantine to the hospital. And the product arrives and is released by QC at about the same time as the patient arrived at the hospital. And just as an aside, these products are also tested for sterility. However, the sterility test goes for about 2 weeks, which means that the product is injected in the patients prior to final QC release, which is the world of radiopharmaceuticals. And you can also imagine with such a tight timelines, it's always just-in-time manufacturing, so we cannot stockpile these products and deliver upon order. They need to be manufactured and delivered as they are made. So it's also right the first time approach, because if there's a hiccup between the delivery of the isotope to CPDC or the delivery to the drug product to the hospital, an hiccup between these 2 steps will result in the rescheduling of the patients. So CPDC offers a wide range of services going from discovery to management of clinical trials, but mostly our activity revolves around GMP manufacturing and distribution of the radiopharmaceuticals and the regulatory affairs. So we have completed over the years over 30 radiopharmaceutical projects. We have designed with our partners 5 radiopharmaceutical facilities, and we are currently supplying over 10 products in clinical trials that are in 15 countries. So we do manage to deliver those products globally. It's a lot of logistics involved, and mostly with iodine and lutetium product for therapeutics. Although we are very active on the global scene, we're also active on the local scene with the delivery of tracers and other drug products across all of Ontario. And we do have ties as well with cyclotron facilities across the country. So if we are to look at our participation in research, clinical research in Ontario over the years, we came to 45 supporting to 45 clinical research trials. And this is by manufacturing, but also with regulatory affairs. In terms of pipeline, we are developing a few products. We do have a commercial product for bone cancer imaging. This product will be transferred to our affiliate CanProbe. And we're also working on 2 projects for imaging agents, the prostate cancer imaging agent PSMA-1007, and the example I showed a bit previously for the CD8 Centyrin. These are the 2 projects that Anne will talk about in her section. And we're also working at producing lutetium out of CPDC in collaboration with Isotopia Molecular Imaging. So we have spun out a few companies, starting with Fusion Pharmaceuticals, spun out of CPDC's R&D in 2017. We have 2 joint ventures, Artemis and CanProbe, for which I will be talking a little bit in the next few slides, as well as New Generis, a company dedicated to the commercial contract manufacturing of radiotherapeutics. So talking about CanProbe, CanProbe is a joint venture between CPDC and UHN, and it really builds upon the strength of both partners. On the CPDC side, we bring our expertise in radiopharmaceuticals and regulatory affairs, as well as our quality system. On the UHN side, it's their infrastructure, access to core facilities, and their extensive expertise in conducting clinical trials. And that venture has been very positive, and we really foresee a great future for this with a very strong implication from both partners. So CanProbe has its own pipeline. We have this theranostic solution for neuroendocrine tumors, the gallium and the lutetium dotatate. The lutetium dotatate is for the therapy, and this program is currently in phase 3, a trial sponsored by UHN, and the product has been licensed out to Point Biopharma. The gallium dotatate for imaging we are currently submitting a new drug submission to Health Canada, and we expect to commercialize it out of CanProbe. And also PSMA-1007 is being manufactured at CanProbe under the sponsorship of CPDC. CanProbe also offers investigational products such as DCF-PYL and other PSMA targeting prostate cancer imaging agent, as well as FASA, F-Colding, and FLT, which are additional cancer imaging agents. We are expecting to expand this offer very soon with 3 brain imaging agents, F-DOPA, PBR6, PBR06, and flutimidamol. So I did mention Artemis. Artemis is a company producing hardware for solid target technology in order to produce gallium and some other isotope. CanProbe is working at establishing this technology in its facility. Just to give you an idea, gallium is one of those with a 1-hour half-life isotope, which means the shelf life is very short. The current technology we use gives us just enough activity to produce 2 doses, which has to be delivered in the GTA because of the small number of activity, the small level of activity. With this technology, we expect to produce 100 times more activity, and with that, having a higher level of products so that we can expand the geographical outreach for our products labeled with gallium. So we do expect to use it in gallium dotatate, but it could be used in other compounds as well, such as, for example, gallium-labeled CXCR4 targeted agent, which is a broad cancer agent. So at this point, Anne will be talking about the OICR projects. Anne?
Okay. Thank you, Bruno, and hello, everybody. The first agent that I'm going to talk about, as Bruno said, is PSMA-1007, which is under development as a prostate cancer imaging agent. Prostate cancer, as you probably know, is the most common malignancy for Canadian men, with 23,000 new cases every year and 4,000 deaths. The overall survival rates are actually quite good if the cancer is localized within the prostate bed, but drops significantly when it becomes metastatic. And unfortunately, 20 to 40% of patients do develop recurrent disease despite curative efforts. Recurrence is detected initially through the rise of the blood marker PSA. And this typically precedes the detection and localization using conventional imaging, using CT and bone scans. So there's definitely a gap in the ability to detect and localize recurrent disease, which then represents a missed opportunity for treating patients early and choosing the right treatment for the patients to give them the best outcomes. Next slide. So this is where PSMA-targeted PET imaging comes in. PSMA is a transmembrane protein overexpressed in prostate cancer cells and is proven to be excellent for the development of both diagnostic and therapeutic radiopharmaceuticals, and has far superior diagnostic performance compared to conventional imaging. F18 label probes have shown very good performance. And as you already heard, we've actually been manufacturing DCF-PYL for a number of years, both for clinical research and a registry study. Next slide. However, we're turning our attention now to PSMA-1007, a new agent with potential advantages over other PET imaging agents. It's already been well documented in the literature in terms of preclinical pharmacology, showing good imaging results, and also tox testing has shown no safety issues. It's been used quite extensively, especially in Europe, in retrospective trials. So over 400 patients have actually received imaging with this agent. No adverse events have been reported. The dosimetry is acceptable. And histopathology has demonstrated that the agent is on target, localizing in areas of PSMA expression. So the big differentiating factor here for PSMA-1007 is the fact that there's very low bladder accumulation. As you can see in the intrapatient images comparing 1007 and DCF-PYL on the right-hand side, there's much less bladder accumulation with 1007. And this demonstrates much better potential for visualizing lesions in the prostate bed and pelvic lymph nodes. Studies have already demonstrated the potential for PSA to impact patient management. Next slide. So as far as Canada is concerned, we're actually lagging behind other countries in that we don't have a commercially available molecular imaging agent to detect recurrent prostate cancer, whereas other countries have metabolic agents such as fluorocholine or FACBC, and now in the US, PSMA-11, a PSMA agent, has now been approved. So we want to change this in Canada. We have licensed PSMA-1007 from the German company ABX and have the rights to develop and distribute the product in Canada. So our goal is to obtain market authorization through conducting a phase 3 study where we'll be looking for an indication for the detection of recurrent prostate cancer. This will improve access to PSMA PET imaging in Canada and facilitate national access to a single agent. Our phase 3 study will have 2 sites, one in London and one in Toronto. And I'd like to acknowledge Glenn Bauman and Metze, who've been instrumental in helping us design the study. Our primary objective is to determine diagnostic performance. We also have secondary objectives looking at safety, detection rate, and impact on patient management. Next slide. So the design is depicted here in brief. After enrollment in the study, the urologist fills out a questionnaire on management. The patient is imaged, the safety follow-up. There's another patient management questionnaire based on the results of the PET imaging. And then 6 months later, we get clinical outcome measures to serve as a composite standard for comparison with the imaging results. And based on that data, we can meet our objectives. So just in the interest of time, I will just say that we have clearance from Health Canada and REB approval. The London site has been initiated, expecting to image patients soon, and PMH is not far behind. Next slide. Okay, I'll change the topic now and just give you a brief outline of our CD8 T-cell imaging agent, an imaging tool for immuno-oncology. As you know, immunotherapy is a game changer in cancer treatment in a number of different cancers, although the response is very heterogeneous with regards to checkpoint inhibitors, for example. Only about 18% of patients get a complete response, whereas the remaining patients only have partial or no response. And furthermore, there's serious adverse reactions that may occur. So it's really important to identify the patients who really are going to benefit from this treatment. Traditional monitoring methods are unreliable. Biopsies, the gold standard, are invasive and prone to sampling errors and not representative of the entire tumor environment or metastases. Radiography is not specific, and it's difficult to differentiate between tumor response and immune filtration, resulting in a phenomenon known as pseudoprogression. So PET imaging has the potential to address this unmet need. And we'll go to the next slide. This can provide rapid whole body imaging, visualizing primary tumors and metastases, and enabling dynamic monitoring of patients. So there's a number of different targets that could be interrogated with PET imaging. We have chosen to look at CD8 expression as a marker of T-cell infiltration. And there's a strong correlation between the presence of CD8 positive T-cells in tumors and the overall survival of patients. So we are anticipating that this could allow for patient stratification and treatment monitoring. Next slide. Our lead candidate is an anti-CD8, known as an anti-CD8 Centyrin. This is an agent that we've actually acquired the rights to from J&J. It's based on their proprietary protein technology known as Centyrin scaffolds, which are based on the fibronectin 3 domain. And these molecules are very small in size, so facilitating good tissue penetration and rapid clearance, which is what you want for a good imaging agent. Furthermore, it's been shown that there's no T-cell activation with the lead agent, and it has very high subnanomolar affinity for CD8. So our goal is to develop a rapid whole body diagnostic imaging tool with specificity for immune filtration. Preliminary in vivo data has been very promising. The labeling of the Centyrin, both with I-124 and zirconium-89, both PET imaging isotopes, has been very successful. It's been safely injected into more than 100 mice and a handful of cynomolgus monkeys. And in rodent models, increased uptake in lymphoid organs was seen in humanized rodent biodistribution studies, and also in cynomolgus monkeys, visualization of lymphoid structures was seen, which actually was reduced in monkeys where there was CD8 depletion. finally, CD8 expressing tumors, xenografts in mice have shown excellent uptake. Next slide. We've actually, whilst those isotopes showed good results, really the isotope of choice is fluorine-18. It's the preferred isotope due to its excellent imaging properties. One can get a robust supply from local cyclotrons with excellent GMP processes already in place, and it's the cheapest option. And there's a range of different kinds of chemistries that can be used to develop the radiolabeling as well. So we've looked at 3 different small molecule linkers. And the approach is just shown on the right-hand side using F18 labeled linker and attaching it to a conjugate of the Centyrin with a second linker in a type of chemistry known as a click chemistry. So next slide, please. And so our preliminary results have shown excellent uptake of the label F18 labeled Centyrin. Regardless of the linker, there's been good uptake in the tumors. However, we've selected the linker Centyrin combination in the middle of the picture here due to the excellent clearance, which will provide good and rapid tumor to background ratios in imaging. So we'll go to the next slide. So as I've said, we had very good tumor uptake and excellent tumor to blood background or ratio of 4.7. And the biodistribution results are shown on the left-hand side of the side of the slide. Furthermore, we've also demonstrated that this product has good radiochemical purity. It doesn't degrade due to radiolysis over a 6-hour period. So it bodes well for developing this agent for the clinic. Next slide. So we've now put together a development plan. We're very busy getting this program going. Our goal is to complete all the development work needed in order to obtain approval for a proof of concept first in human clinical trial. So we're doing a bit more optimization of the chemistry, and that'll be followed by some further biological validation. We're also looking into the scaling up of linker manufacture and getting scaled up supplies of Centyrin in place. We will meet with Health Canada to talk about our plans, and following that, we'll conduct toxicity testing and develop our GMP manufacturing processes. With the ultimate goal of getting a CTA submission cleared by Health Canada to start the proof of concept phase 1 clinical trial. So that's the end of our presentation. I'd just like to acknowledge both our collaborators and our colleagues